Hi everyone. Good afternoon. My name is Clara, and I am from UCSD Dr. Trey Edekers Lab. And today, it's really my pleasure to talk about my recent work titled "Evaluation of Large Language Models for Discovery of Gene Set Function." So first of all, I want to first provide you some background of omics research. So one of the goals of omics research is to identify clusters of genes that work coherently towards similar biological functions. For example, here showing in the heat map that uh, here showing on the heat map that you can identify sets of genes with similar expression pattern and suggesting that they might have similar biological functions across different conditions. And similarly, proteomics research do experiments like co-abundance or the measurement of physical interactions further identify proteins that work closely with similar functions. But as we identify those clusters of genes or proteins, what should we do next? How do we interpret those clusters? So here I'm showing an example of 16-member gene set that derives from protein-protein interactions. How should we attempt to interpret it? When I say interpretation, I'm more specifically, I want to ask question like, what are the function or functions of each genes? And is there any evidence that they work coherently or they belong to a coherent cluster? In addition, do I really believe that they work similarly? Or if yes, then how do I name this cluster? What is the best description of this gene cluster? And as I'm doing this analysis, can I provide a succinct report for all those to summarize all these approaches I did? So in order to check all those boxes, it means that you need to spend a lot of time, even a lot of labor, especially if we want to scale it up with a bunch of gene sets, or if the gene set is really big. So I believe a lot of you in the audience have already tried to chat with ChatGPT, and the first talk this afternoon have talked about how using large language models can help us retrieve Go terms. And so you might be, for me, I'm really impressed by the text summarization ability and knowledge it has, GPT-4 has. So as omics researcher, we are kind of curious to see if LLMs can actually help us synthesize the gene set function. It's basically help us with the interpretation that I was mentioning in earlier slides. So to test it out, we just quickly open up ChatGPT and ask simple question, paste in the gene set and ask, can you tell me about the functions of this gene list? And can you provide me a name for the gene list and give me a report of your analysis? So just by this simple trial, you can already see that it can generate a summary paragraph, a summary essay for those genes. It describes all the functions of the genes that involved in this cluster, and it gives a where's my cursor? It gives a summary paragraph of the coherent function that it believes they work together. However, there's still some clear defects with this simple approach, is that the name is overly broad. Like everything is involved in cellular regulation. The name doesn't really tell us about the biology behind these gene clusters. And even though I truncated the text, but the full text is kind of verbose. It repeats itself a lot. So this means that, but nevertheless, we can see the opportunity that we can use LLM as an assistant for to assist us in interpreting gene set functions. But we might need some careful prompting to guide the LLM towards our goal. So with that in mind, I want to bring your attention to our engineered prompt that we focused on using some effective prompting strategies to guide the LLM more precisely. And now I want to break down each part of the prompt. So first, we start with specify a role for the LLM. Doing that can help us narrowing down the specific fields that we want the LLM to focus on. So here we want it to be focused more on the molecular biology. So we told it to be an assistant for molecular biologist. Then we followed a pretty popular technique called chain of thought prompting. So this is the prompting strategy that guide the LLM step by step through the reasoning process to help the LLM understand the task better. And research has shown that doing so can help the LLM with solving the problem and provide output that's more aligning with your expectation. For example, we ask it to create a critical analysis for the system of genes, and after doing your analysis, then propose a name that can be descriptive of the biological processes. And we also introduce a concept called confidence score to the LLM. So we ask it to provide a confidence score. We told it what it means by 0 and 1, meaning the lowest and highest. And we give a definition of confidence, which is how accurate the chosen name is representing the functions of this cluster. So you might ask why it is important to have the confidence score. So this additional layer of letting the LLM self-assess its performance can help us, can give us a hint of how much we can trust the LLM, because we ask it to express its uncertainties. And I will bring this back later when I show the result. And more importantly, I also ask the LLM to not naming a gene cluster if it cannot find any coherent functions within the cluster. So why it is important not naming a cluster? So in real life scenarios, especially for omics research, you might it is possible that we can have artifacts, contaminations, or even false discoveries throughout the process of experimentation or downstream analysis pipelines. So it will be helpful if we can get a hint of potential noise in our data. And also, we don't want the LLM to hallucinate the names for gene set just for giving a name for any random things. Then further refining, within the prompt, we further refine the model's output by additional guidelines that being concise, factual, specific. And we follow another strategy in our prompt. It's called few-shot prompting. And one-shot is a subset of few-shot prompting. So few-shot prompting is to give my model a few examples so that it can understand the desired format or how it can approach to solve the problem. And research such as this paper that I pasted have shown that it really helps with the LLM in understanding the question and solving the task. And so this is again example that we give it a list of proteins. We give the name and the confidence score. We as human, how we would describe the function or roles of genes. If we saw genes that work similarly, then we group them together in our summary paragraphs. And at the end, we provide a conclusion or discussion paragraph summarizing why we think those genes work coherently or not. And lastly, in the prompt, we give the list of genes. So to wrap up, applying this prompt, we then develop an automated pipeline to input the gene set, use our prompt to ask different LLMs, and we organize the output to its name, analysis, and confidence score. And we wrap this pipeline into a online web portal. We call it GSAI. And here I would like to show a small demo of how we use this web portal. I just paste in the gene set I showed earlier slides, and I select which model I want to use. I click run query, and it's starting to do its analysis real time. And what you can also do is to click show prompt, and you can see what is our prompt. You can copy it to ask your interactive question in ChatGPT or other models of your interest, and can copy the results if you want to save it somewhere so that you can review in the future. And the overall layout is it start with the gene set name, the confidence score, and the analysis essay. Okay. So now, as we already built this pipeline, that we can automate this task, that it's being helpful for omics researcher. But as scientists, we might raise further question: is that how can we evaluate the results from the LLM? How do we know it is good at this, and how can we trust the response from the LLM? So that being said, towards that aim, we build a pipeline for evaluate the LLM's response. And our first approach is to evaluate whether the LLM can recover known knowledge. So to do that, we benchmark its output against canonical knowledge from gene ontology. And I believe a lot of you have already know that gene ontology is the database of gene sets with human curated names. So we gave the LLM the same gene set from Go and evaluate how much the names they propose aligns with the known knowledge that is the Go term name. And the metric we use is called semantic similarity. So this evaluates the conceptual closeness between words or phrases, not just the exact match. So what we can see is that most of the LLMs can do pretty good job in returning similar names that is already known in the database, except the Llama 270B. And the take-home message is that LLM has its ability to recover known information that's potentially embedded in their training data. Now I want to move forward to bring your focus to the confidence score here. So we ask it to express its uncertainty, but we also want to evaluate whether we can trust its uncertainty. So we build a pipeline to assess the LLM's confidence by randomizing the gene set. So we take the term from Go and randomly inject random contaminations to the Go gene set, or completely randomize the gene set to test if using the confidence score we can differentiate between those different sets. And here is a result. I want to remind you that I've asked in our prompt, we asked the model not to name a gene set, which is shown as the gray bar here in the plot. And you can already see that GPT 3.5, Mengzhou Instruct, and Llama 270B don't feel comfortable with refusing a name. In other words, they always give a name, even though it is a random gene set. So that is the output that we don't really want, because it means like it as either proposing something that's really bad, like for example, every gene's involved in the cell or something, or it is hallucinating because it's trying to give a name for the gene set. So we're narrowing down to the two models, that's GPT-4 and Gemini Pro. They can refuse names for the random gene set. And we also look at the common approach as functional enrichment analysis. It is the statistical approach that match the gene set of interest with what is inside of the knowledge base. And it returns relevant biological processes or functions. So the number of unnamed random terms are pretty similar between GPT-4 and the statistical approach, whereas Gemini Pro is is a little bit off. And in addition, GPT-4 refused more in the contaminated sets and gives lower score confidence for contamination. So now we have shown that the LLMs can self-evaluate and they can recover known knowledge. We want to see how it applies in real life scenarios in the omics gene set that we derive from experiment. So we take a pool from 300 gene sets that derive from differential expression from protein interaction experiments. This means that we do not guarantee there's prior knowledge of those gene sets. So we want to test how LLM can perform in such more practical utility. So we saw that GPT-4 can propose names with confidence in 135 cases. And I want to bring your attention to their analysis of this example. So the GPT-4 gives the name regulation of colon ring ubiquitin ligase complex for this 16-member gene set. And as we reading through the essay, you can see it lists out functions for each genes or each subset of those genes. I'm showing the truncated paragraphs, but the full paragraph as described gives detailed description of each gene. So it's being more inclusive in their analysis and showing what those genes are functioning in. And in their summary paragraphs, they explained why they think those genes should be categorized in this in this function. And they also show that they made a conjecture for the Wnt kinases that they are not directly involved, but it is possible that they do such it's not a direct involvement of the function. And I want to note that we have gathered a panel of human scientists to actually go into those analysis paragraphs and manually verify the facts that those models generated, and the majority of them are actually verifiable. So I want to summarize my talk that we've built a pipeline to use LLMs to do gene set analysis and recapitulate known knowledge, and we evaluated its self-assessment ability, and we really value its analysis essay. It brings a lot of value to practical use cases. And with that, I would like to thank all the co-authors on this paper, and especially Dexter Pratt, who's in the audience. He brings a lot of help with the brainstorming and all along this project. And obviously, my advisor, Trey Edgar, for supporting this project. And feel free to scan those QR codes for the paper. Because of time limit, I didn't include all the results, but feel free to check out the paper and the website. And if you test out the website and so see some and you have some feedbacks, feel free to reach out to us. Thanks.
Thank you.
So that's excellent, and I really thank you for developing this. As someone who's done a lot of PubMed gazing in my time. Is this on? Can you guys hear back there?
Can you guys hear in the back?
So my question is citations. I'm not asking you to, write a paper for me, but if I had citations with such a summary.
Yes, I'm really thank you for asking this question. We have to think about it, and actually, in our following pipeline, we search PubMed for citations, and we attach those references along with the analysis paragraphs. So we did using a automated pipeline to give us how to validate those statements, and you can check out the paper. It's in the paper. Yeah. Thanks.